Welcome friends. Now we are going to learn about checkpoints in cell cycle. Checkpoint in cell cycle. What are checkpoints? There are following different checkpoints in cell cycle. Three different checkpoints are there in cell cycle: G1 checkpoint, G2 checkpoint, and G3 checkpoint. Okay, let's see what are the checkpoints. What are the checkpoint? Checkpoint means what? These are the particular point. Checkpoints are the particular point in the cell cycle where cell evaluate itself whether she is ready to divide or not. Okay, because in cell cycle, during cell cycle, cell is going to divide and for production of new cells. Okay, and during this process there are some checkpoints. Cell evaluates itself whether she is ready to divide or not. That is why. So clear. Checkpoint means what? It is the particular point in the cell cycle where cell evaluates itself whether she is ready to divide or ready to enter in next phase or not. That is nothing but the checkpoints, and such a three checkpoints are there in cell cycle, which is known as G1 checkpoint, G2 checkpoint, and G3 checkpoint. Let's see what is G1 checkpoint, where it is occurrence of G1 checkpoint. It occurs at the end of G1 phase and beginning of the S phase. Okay, where this G1 checkpoint is it occurs at the end of G1 phase and beginning of S phase. Okay, at the end of G1 and beginning of S phase. What it checks, what cells evaluate during this G1 checkpoint. So during this G1 checkpoint, cell evaluates whether the all necessary components which are required for the synthesis of DNA is successfully gathered or not. Because after G1, when this G1 checkpoint starts, it comes at the end of G1 phase. Means now it is cell is entering in the S phase. After G1 there is S phase. And in S phase we know that in S phase there is the synthesis of DNA takes place. Okay, and for synthesis of DNA there are various components which are required, such as proteins, RNA, and different enzymes. They are required for the synthesis of DNA. Before entering S phase, cell wants to evaluate that whether she is ready to enter in S phase or not, because after entering in S phase, cell have to produce or synthesize DNA. Okay, and for that she needs protein, enzymes, and RNA molecules. Okay, so this checkpoint checks whether all necessary components which are required for the synthesis of DNA are successfully gathered or not. Okay, or it gives the assurity of successful synthesis of all the components which are required for the DNA synthesis. Okay, here next move towards the G2 checkpoint, where it is occur at the end of G2 phase and the beginning of M phase. Means G2 checkpoint occurs at the end of the G2 phase and beginning of the M phase. And what it checks? It checks, first of all, it checks whether the complete and proper set of the DNA is successfully synthesized or not, because during S phase there is the synthesis of DNA is done. Okay, suppose human chromosome number is 23. Human DNA has 23 pair of chromosome number. Okay, if any one pair of human chromosome is missing, it will lead to the abnormality in human genome. Right? So that it checks, this G2 checkpoint is there for the checking of whether the proper and successful DNA synthesis is there or not. Okay, its first job is to check whether the complete and proper set of the DNA is synthesized or not. If a complete and proper DNA is synthesized, then and then only it will allow to enter in next phase. Well, one more thing is I missed to tell you that during G1 checkpoint, if anything, suppose here for the synthesis of DNA, we required enzymes, proteins, and RNA molecules. If any compound among these is missing, at that time cell will block cell division there. a cell has ability to block cell division at any stage. Okay, and this is the stage where cell can stop, or cell can stop cell division. Okay, means if any compound is missing or any compound is remain to gather, then cell will block cell division. Means cell has complete control over the cell division. Whenever cell wants to stop cell division, cell can stop cell division. Cell has the ability to stop cell division. If cell loses this control over the cell division, if cell loses, what will happen? That cell will become the cancerous cell, and there will be the formation of tumor or lesion, because in cancer cell there is uncontrolled multiplication is occur. Huh? And when this uncontrolled multiplication is occur, if when cell loses its control over the cell division. Okay, here cell is in its complete control. Okay, let's move towards the DNA. First, in G2 checkpoint, what it checks? It checks whether the complete and proper set of DNA is synthesized or not. Next, this G2 checkpoint have to check that whether all necessary cell organelles like mitochondria, chloroplast, different organelles are successfully synthesized or not, because these are the very important organelles. Like, because in animal cell mitochondria is powerhouse of cell, and in case of plant cell chloroplast is the energy generating organ. Okay, site of photosynthesis. Okay, so therefore these organelles must have in a cell. That is why it ensures that whether these cell organelles are successfully synthesized or not. If anyone is not synthesized, then this G2 checkpoint will not allow to enter in next phase, that is mitotic phase. Cell division will block there. Okay, another what it checks? Third, it checks whether some fibrous protein which are required during the actual cell division or for the formation of spindle fiber in the mitotic division, that also synthesized here, and different proteins which are required for the synthesis of cytoplasm, that is also synthesized. And also it checks whether the sufficient amount of cytoplasm and nucleoplasm is synthesized or not, because from one cell there is for formation of two daughter cells are there. Okay, there will be formation of two daughter cells, and for that two daughter cells will be identical to their parents if they divide mitotically, or they have all necessary organelles including nucleus, mitochondria, cytoplasm, nucleoplasm, and they are exact replica. They will be exact replica of their parents, so that everything should be there. If anything found missing, this G2 checkpoint will not allow to enter the cell in next phase, or cell division will be blocked there. That is why these checkpoints are there, because at that point cell evaluates whether everything is proper, we are ready to enter in next phase or not. If anything is missing, cell will stop and cell will regenerate that or resynthesize that particular cell organelle, cytoplasm, etc., and then then only cell will move towards the next phase. Let's move towards this G3 checkpoint. What is G3 checkpoint and where it occurs? It occurs at the metaphase. In mitotic phase, there are various phases: prophase, metaphase, anaphase, and telophase. In that metaphase, during that metaphase, this G3 checkpoint is occurs. And what this G3 checkpoint checks? It checks whether during my metaphase what happens, because after metaphase there is anaphase, and in anaphase there is separation of spindle fibers, are the separation of chromatids will be there, and that for that separation spindle fibers get attached to the centromere of the chromatids, and after the contradiction of that spindle fiber, two chromatids separates and they acquire polar region. Okay, and that binding, here G3 checkpoint checks whether that spindle fiber is successfully attached to the centromere of chromatids or not. If attachment is not proper, what will happen? Separation will not be proper. In anaphase, it will create problem. Okay, therefore cell will block cell division there. Okay, its role is to check whether the spindle fibers are properly attached to the centromere or not. If spindle fibers are properly attached, then and then only it will allow to enter in the next phase, otherwise cell division will be blocked there. Okay, this is what the cell checkpoints of cell cycle. Okay, thank you. Please like and subscribe my channel, and if you have any question, please comment me, please mention it in comment box. Thank you.